Welcome to the Honig Lab PrePPI website. In this video, we'll be discussing the interaction structure and table panels on the website's results page. We'll be using the gene AP1G1, which corresponds to the UniProt ID 043747 as our example case. We'll begin with the interaction table on the right-hand side. Each row in this table represents a predicted or known interaction partner for the query protein. The table includes the partner, UniProt ID, gene name, PrePPI scores, and any supporting experimental database evidences. The table can be sorted by clicking on the column headers as well. In the PrePPI SM score column, a drop-down arrow appears when a structure-based score is available. Clicking this drop-down reveals the PDB template that were used for the structure-based scoring and model building for that interaction. Similar to the PrePPI SM score, the PrePPI Slim score column also includes a drop-down menu when a score is available. Expanding this drop-down provides additional information about the peptide and the protein domain involved in the predicted interaction. When available, the drop-down also includes a link to a visualization page, where users can explore the PrePPI structural templates associated with the peptide domain interaction. Clicking on a PrePPI complex will select the complex for viewing. For further viewing functionalities, please refer to the MoldStar RCSB documentation. Hovering over the amino acids will tell you which chain the amino acid is part of. For example, here, this amino acid is part of chain A, and the red colored are part of chain B. These are related to the protein receptor domain for chain A, and peptide is chain B. The database entry column also indicates whether a given interaction has been previously reported in an experimental interaction database. If a protein-protein interaction has an experimentally defined structure in the protein databank, the PDB, will appear in this column, along with a direct link to a corresponding PDB entry. This makes it easy to distinguish between purely predicted interactions and those supported by existing experimental structural data. On the left-hand side of the page is the Interaction Structure Viewer, which displays the three-dimensional model for the currently selected protein-protein interaction. Clicking on a partner in the interaction table is required to load and display the interaction model. Once a row is selected, the corresponding interaction structure is shown in the viewer on the left. At the top of the viewer are three display options: View Model, View Template, and View Superposition. View Model displays the predicted interaction structure generated by PrePPI. View Template shows the experimentally determined PDB structure that was used as the template to build the interaction model. View Superposition overlays the predicted model onto the template structure, allowing users to directly compare the prediction with its underlying structural reference. The structure viewer is fully interactive. Users can rotate the structure by left-clicking and dragging, zoom using the scroll wheel, and explore the interaction geometry from different orientations. Beneath the structure viewer is the View Interaction Pair button. Clicking this button navigates to the protein-protein interaction pair page corresponding to the selected interaction, where additional details specific to that interaction are presented. There are also structure download options. Users can download the interaction model, the template structure, or the superposed structure as PDB files for use in external molecular visualization or analysis tools.